Hello and welcome to this podcast. In this podcast, we're going to talk about protein folding. The importance of it, how it happens, what happens when it doesn't fold properly. We're going to talk about how we have this linear piece of protein and how this will eventually fold up into a mature protein. As we begin this discussion, I want to talk about 4 important bonds that occur to allow us to go from this linear structure to this fully folded protein. The first are disulfide bonds. Disulfide bonds occur between 2 sulfur groups. So if you remember from the previous podcasts, there were only 2 amino acids that had sulfurs in them, cysteines and methionines. However, only cysteines will form these disulfide bonds. The next bond that I want you to keep in mind are hydrogen bonds. Hydrogen bonds, as we form these structures here, are going to be very important in both these intermediate steps and the final structure here. And the amino acids that are going to be important in hydrogen bonds are going to be the polar amino acids. So any of the polar amino acids could participate in hydrogen bonding. Electrostatic attractions. The amino acids that are going to play a role in this are going to be the positive and negative amino acids. So for the positively charged ones, we're talking about lysine, arginine, and histidine. And the negative ones, we're talking about aspartic acid and glutamic acid. The fourth type of interaction that's going to be important in going from this stretch of amino acids into this fully folded protein are van der Waals. And remember, these van der Waals occur because of these very temporary positive and negative charges that exist because of the way the electrons spin around the outer shell of the atom. And lastly are hydrophobic interactions. The tendency to get those hydrophobic residues, rather the nonpolar ones, into the middle of the protein and allow the water in the aqueous solution to interact with these polar residues. Now, all these bonds are important in forming the structure, but there are 2 factors that are important in governing how these amino acids will be placed next to each other to allow for these interactions to occur. The first is what we call the primary structure. So right up here, I wrote this out in a linear form where you have the first amino acid, the second, the third, the fourth, the fifth, the sixth, and so on. We call that the primary structure. We'll talk about the levels of structures in a little bit, but for now, that's the primary structure. How these amino acids appear play a critical role in eventually how they're going to form this fully folded protein. The other thing that's important are energy considerations. What I mean by energy considerations is that this process of going from this primary structure to this fully folded form here, this whole series of steps are energetically favorable. The overall delta G is negative. In the process, it releases heat. To create this great order here, it has to allow that heat to be released, if you remember from before, so that the entire system still moves towards disorder. And because of these 2 factors, the primary structure and energy considerations, this allows certain amino acids to be next to each other to allow those bonds to form that we talked about on the last whiteboard. As scientists began to study protein folding, one of the big questions that popped up was, can proteins fold spontaneously? Now, there was this one scientist who did a very elegant experiment. His name is Anfinsen. You don't need to remember his name, but he was the one that did this experiment, and it's a really important experiment. He took an enzyme called RNase A, and so let me just draw it like so. And this RNase A enzyme was stabilized by various disulfide bonds, as well as some other forces such as hydrogen bonds and electrostatic interactions. I should also mention that RNase A, its job is to degrade RNA. He took this enzyme and added a reducing agent, 2-beta-mercaptoethanol and urea, and that helped break these disulfide bonds. And what happened then was the protein unfolded, and it's also non-functional at this state. What he discovered was when he removed these chemicals, so no 2-beta-mercaptoethanol or urea, unfolded protein here would fold back and be functional. This suggested that this protein, this enzyme, could fold spontaneously, and indeed it can fold spontaneously. Now it's important to note though that even though Atkinson showed that this enzyme and others showed that other enzymes can fold spontaneously, we now know that the folding of the protein often requires helper proteins. So let's talk about those next. So to answer this question, can proteins fold spontaneously? Let's just write sometimes. I just mentioned that RNase A could fold spontaneously without the help of other proteins, but sometimes there are other proteins which require help. And there's a whole family of proteins called chaperones, and chaperones assist in the folding of some proteins. Really, I should probably say most proteins, because most proteins do seem to require some help here. And even the ones that can fold spontaneously, we often find in physiological conditions, they often will use chaperone type proteins. So what I'm going to do here is I'm going to draw a ribosome, as lovely as it is here, and this ribosome will have a messenger RNA being fed through it. And as this messenger RNA is being translated into protein, we see protein coming off the side here. Chaperone proteins will often just immediately bind to these other proteins and help fold them. They will position the growing amino acid chain here, the growing protein chain here, in such a way that encourages the right folding, the correct folding of that protein. So as this protein is coming off here, these chaperone proteins bind to specific sites there and help assist in the final folding of this protein. And then the chaperones are released to go back here and redo the job with new proteins being made. In other instances, you could have a chaperone that acts like a chamber. I'll draw it like so. Proteins that are being made before they get folded, and before they get folded incorrectly, are taken inside of this chamber. And inside of this chamber, they're locked in here, so to speak. And in the chamber, because the area is much smaller, it encourages the right folding of this protein. So eventually it will fold in a correct form and then be released from this chaperone protein. Now I've alluded to this before, but I want to talk about the levels of protein folding. The first one is called primary. What the primary structure is just that linear sequence of amino acids. So from the N terminus to the C terminus, you have one amino acid, two amino acids, three amino acids, and all the way down until you get to the N. As we've discussed before, each of these amino acids are held together by peptide bonds. So we would say that the primary structure is characterized by peptide bonds. It is also a non-functional structure, meaning that for that protein to have function, it has to go beyond this primary, this linear array of the amino acids. It needs to fold more. But ultimately, the sequence of the amino acids in this primary structure ultimately dictate how that protein will ultimately fold in its final conformation. The next level is secondary. There are two main kinds of secondary structures. I'm going to write them here: alpha helix and the beta sheet. Both of these secondary structures take advantage of hydrogen bonding between amino acids that are near them. Not necessarily right next to them, but in the vicinity of each other. We could draw an alpha helix like this. It forms just a simple helical structure. And again, it is characterized by hydrogen bonds between amino acids that are relatively near. So this alpha helix is often found in membranes, or membrane proteins I should say, that function in transport. Beta sheets can be drawn sort of like this, where they have these distinct edges to them, like so. And they're connected with each other by these linker pieces of amino acids, like so. Like the alpha helix, they're characterized by hydrogen bonds. Again, local interactions to form these kind of steps here. But they will also form hydrogen bonds between different beta sheets. These structures that are formed, sometimes when they're stacked like this, we call them beta pleated sheets. And what they do is they add a lot of rigidity for the entire protein, or at least for that part of the protein. They also often form the core of the protein. They're very stable units. If you think about silk fibers get their really unique tensile strength because of beta pleated sheets like this. I'm going to erase this now so we can talk about the next level of protein structure. The next level of protein structure is the tertiary level. Remember, you had your primary level here, and this primary structure then formed alpha helices, maybe some beta sheets, and most of these interactions occur due to the attractions or the bonding of amino acids in the relative area. As we move to the tertiary structure, these secondary structures will now fold upon each other, and there's often several of them. Maybe more alpha helices. Maybe we're going to get a beta sheet here that'll form on itself. Something on these lines here. And these are held together by a variety of bonds, including hydrogen bonds. In fact, almost everything here. Van der Waals, electrostatic, and perhaps the most important, it's hard to say one's more important than the other, but this is really critical for the folding of this tertiary level of the protein. And that are these hydrophobic interactions, where the necessity to place these nonpolar amino acids in the middle of this protein, due to the fact that the aqueous solution around here is interacting with these polar residues, pushing those nonpolar residues into the middle of the protein. Often, these are stabilized by cofactors. Cofactors are often metals like zinc, iron, copper that will bind here, and they will stabilize these interactions to produce the most stable structure of that folded protein. Other factors to think about, so these are cofactors that can bind, but the ultimate folding or unfolding of this protein can also be influenced by pH, temperature, and salt. At the right concentrations or values here, they help stabilize this structure. Changing these can help destabilize this structure. Typically, the tertiary level is the functional level. The last level here that I want to talk about is the quaternary structure. And what this is when you have one tertiary structure like this one here, or we might call it a subunit, would be a better term, interacts with another subunit over here. They can be the exact same protein as over here. It could be a different protein. So here I'm going to put subunit 2, and over here is subunit 1, because they're different looking at least, right? When we have two interacting with each other, we say that is the quaternary structure. When it's two, more specifically, we would call that a dimer. If there are three, we call it a trimer, 4, a tetramer, and so forth. If they're different, like we see here, they would be called heterodimer, heterotrimer, heterotetramer. If they're the same, we would call them homodimers, or homotrimers, or homotetramers. These quaternary structures are also stabilized by these same kinds of interactions, and they are also considered a functional form of the protein. Some proteins only need a single subunit to function, whereas others require 2 or more subunits to function. The next thing I want to talk about are protein domains and these intrinsically disordered sequences. Let's start with the intrinsically disordered sequences. If you remember, I mentioned these as linker amino acids. And so they exist. Maybe you have an alpha helix here, and then maybe you have a beta pleated sheet over here, and maybe another alpha helix here. Sometimes there are regions without structure, without a secondary structure, like here. And their purpose, as far as we can tell, is to stabilize this structure, to make sure that these secondary structures are held in the right position. At one time, we didn't know they existed, because when you do experiments on these, often these regions here are the least protected, and they're easily degraded during experimentations with them. In addition, these unstructured regions don't form crystals very well, so when scientists did X-ray crystallography studies on these proteins, these typically were not part of that final structure they determined. Now, there can be also these unstructured regions in other parts of the protein that are not here. Or these intrinsically disordered sequences could also not be associated with linkers, but they could just exist independently, like so. They can do lots of things, and they're responsible for several things within a protein. They add flexibility, the ability to bend. Because of their flexibility, they can wrap around other proteins or substrates. The next thing I want to talk about are protein domains. So to talk about this, I'm going to draw different structures of a protein, and I'm not going to draw the specific secondary structures this time. I'm just going to draw them as kind of these globs. Now, I drew them in 3 distinct colors here, even though this is a single protein that I drew. One protein. But this protein has 3 domains. So it has 3 domains. Let me write that here. Each domain behaves independently. They can fold independently of the other 2. They can have different catalytic abilities or purposes. For instance, maybe this purple domain has a DNA binding function. Maybe this domain has a substrate binding, substrate other than the DNA. Maybe it's a substrate that binds here and it directs it to the to a different location in the cell. And maybe this middle one here, once it's bound to the cell, the DNA or somewhere else in the cell, it has a 3rd catalytic function. I want to remind you that since this is a single protein, it's a single gene that codes for it. One gene with one start, one stop, makes one big protein. They each fold independently, as shown here. They each have independent functions, though they're related. This binds the DNA, this binds a substrate, say, to take it to the nucleus so it can bind the DNA, and maybe this has some catalytic function in like transcription or replication. Having these different domains aids in evolution. It's much easier to work from a gene that already works, as opposed to trying to make a brand new gene. So if this gene is duplicated somewhere else in the genome, you've maintained this original copy and it keeps doing its job. But this one here can change a little. Maybe there's going to be some sort of advantage to having another DNA binding protein, but maybe it's going to bind a different substrate. Maybe, and this is just a fictitious protein here, but maybe this one here bound sucrose, the sugar sucrose. So let me write that one here. Maybe the environment changes and such where it's going to be more advantageous to have a gene that can also bind lactose. You still have the ability to bind DNA because you haven't changed this part of the gene. It still has its central catalytic function, but now maybe it will be regulated by binding a different substrate. Maybe we want a 3rd set here, and we don't need that one to bind DNA. Maybe we want it to have some other function where it binds sucrose and has a new function in the cytoplasm, for instance. By having these different domains, it allows to maintain very specific parts of it while changing other parts of the the gene and ultimately the protein. Now this brings us to a new topic, and I want to keep this all here because it's related, and that is protein family. As these new genes are being formed through mutation and duplications, they maintain enough of the sequence here, enough of the original function, that we say they form a protein family. You can have protein families that will have similar functions. They'll have similar sequences. And these family proteins can be within a species or between species. You can think of like the globin family. The globin family, within, say, humans, there are many different proteins that we call globins, and they're in the same family. But those same globins are found in other species as well. And they have similar functions, similar sequences. Okay, that ends all I want to say right now in this podcast. We're going to spend more classes talking about protein, so we're not quite done yet. But up to this point, what we've talked about were amino acids, how they're classified based on those 4 categories of nonpolar, polar charged, positive or negative, or polar uncharged. We talked about how they are linked via a peptide bond. And we talked about how the sequence of the amino acids via a peptide bond to form a protein, and how these proteins are very well conserved within a species and between species. Next, we talked about protein folding. We talked about the important bonds that were involved. We talked about how it occurs. Either it can be spontaneous, like with the RNase A, or it can need the assistance of chaperone proteins. We talked about the 4 levels of protein folding. We talked about functional domains, and we ended by talking about protein families. Okay, that is all I have for right now. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.